Hello and welcome to your first video lecture for pathophysiology. In chapter 1, we're going to talk about cellular biology, which covers a very broad spectrum of topics. I would say this is going to include a lot of information that you learned, I would say, within the first third of your AMP1 course and a little bit of microbiology. So with that said, let's go ahead and get started. The first thing we want to know is the differences between prokaryotes and eukaryotes. So our prokaryotes have no distinct nucleus. They're going to have their genetic information kind of floating in the middle somewhere in an area that we call the nucleoid, and it's singular, circular as far as its chromosome. We're also going to see a lack of histones and other organelles. We might have some. We've got these ribosomes here, the cytoplasm, but we really don't have those membrane-bound organelles that we see in eukaryotes. Examples of prokaryotes would be like our cyanobacteria, bacteria in general, and rickettsia. As far as our eukaryotes, these are more complex cellular organizations. We'll have our membrane-bound organelles here, such as our mitochondria, and we see a well-defined nucleus with several chromosomes in here. And examples of our eukaryotes would be higher animals, plants, fungi, protozoa, and our algae. And within our eukaryotes, we're going to see some processes like protein synthesis, transport across our outer cell membrane. We'll get into more of that in a few slides, and then our enzyme content is higher. So let's talk about those cellular functions a little bit more. We know that they're going to be specialized through differentiation or maturation of these cells. And first up is movement. When we think of movement, the first thing we're going to think of really are our muscles, because we know they're attached to our bones and they produce movement. We also know we find muscle within our hollow tubes like our arteries and our intestines, and other cavities can be lined with muscles too that can help move or empty contents from a structure as they empty, such as our colon. Next, we have conductivity. And when we use the word conduction, we're discussing a response to a stimulus. And this is done through a wave of excitation, and an electrical potential is going to pass along the surface of the cell to reach other parts. So when we think about our neurons, our nerve cells that send that impulse through the axon, that's the ability to have that conductivity. Next up is metabolic absorption. All of our cells can take in and use nutrients and other substances from their surroundings. Simple enough. Then we have secretion. So this is when we have certain cells, like a mucus gland cell, let's say within our intestines, that can synthesize a new substance from substances that they're absorbing. And then they secrete that new substance so that they can serve as needed elsewhere in the body. So I mentioned the mucus cells that we have within our intestines. They're secreting mucus into the lumen so that they can kind of help the food in there compact and without friction move through the intestines. Next, we have excretion, and all of our cells can get rid of waste products. And as we are getting nutrients, we're going to get some metabolic breakdown left over, right? So we can use certain organelles such as lysosomes that are going to contain enzymes to break down and digest those contents and then eventually rid them from the cell. Respiration is also very important. We know our cells are going to absorb oxygen, and that is used to transform our nutrients into energy, which we know as ATP. So later on, we'll talk about cellular respiration or oxidation, and of course that happens within the mitochondria. Next up is reproduction. And in reproduction, we're going to have tissue growth as our cells enlarge and reproduce themselves. And even without growth, we have some tissue maintenance that's going on, right? And that's going to require that our new cells be produced to replace the cells that are lost normally through cellular death. And not all of our cells are capable of continuous division, though. And lastly, we have communication. We're going to get into specifics regarding that as well, but just know that having that communication for our cells is going to help them survive as a society of cells. And also, it allows a maintenance of a dynamic steady state. All right, a quick question here to test your knowledge. A cellular function that occurs in all cell types is... Now, we just mentioned that reproduction was not one of those, right? That was one of the last ones. So if you came to the conclusion that D, metabolic absorption, occurs in all cells, you are correct. We're taking in those nutrients and other substances nearby our cells. We have movement, but we also know that that is not performed by all cells. Muscle cells are primarily the ones that help us do that. And secretion is not done by all cells, but by certain cells like the mucus glands that I mentioned earlier. So next up, we're going to get into all the specifics of the eukaryotic cell. A lot of this should be review for you, so pat yourself on the back if you remember all of this. We know that this cell is going to consist of a plasma membrane. So let me back up so that we have a picture of our full cell. So here we see the plasma membrane surrounding the entire cell. And then we've got our cytoplasm, which is really going to be that fluid-filled area of the cell between the organelles and the nucleus. And then we have our intra, meaning inside, cellular organelles. So first we'll discuss the nucleus, which we see pictured over here and here. And this is our largest membrane-bound organelle and will assist us with cell division and genetic information. We'll talk about cell division toward the end of this lecture, and the genetic information will be touched upon more in Chapter 2. So our cytoplasm, we said, was this cytoplasmic matrix that we find within the cell. It's filling up that space really between the plasma membrane, having these organelles suspended within it, and then all the way up to the nucleus. Now that fluid portion is the cytosol. And as far as its function, it's really going to help certain cellular processes take place that include metabolic reactions. It also supports and suspends our cytoplasmic organelles that we mentioned. It helps maintain the cell's shape and facilitates movement of molecules within the cell. This is especially true when we go through cell division. Next up, we have our ribosomes, and these are RNA protein complexes that are synthesized within the nucleolus. So if you remember our nucleus, in the center of that, kind of like a core, is going to be the nucleolus where these ribosomes are made. And this is going to also help us in the process of cellular protein synthesis. If you remember, it kind of clamps down on that mRNA and pulls in the tRNAs in order to attach all the amino acids to one another. Another organelle is going to be our endoplasmic reticulum. And if you recall, we have a rough one, which you could see pictured over here with all of the ribosomes attached to it, and a smooth ER that does not have those ribosomes. They do create these spaces known as cisternae, and their role is to really synthesize, fold, and transport proteins and lipids. We do see that the smooth ER is more involved in the lipid process, whereas the rough ER is more involved in the protein process. Then we have our Golgi complex. So this is extended out a little further into the cytoplasm and is a network of these smooth membranes. They are used for processing and packaging proteins. So essentially the rough ER here goes through, folds them, prepares them, and then sends them over to the Golgi apparatus. through secretory vesicles. And once they move through the Golgi apparatus, these proteins will also be set into secretory vesicles and sent either to other areas of the cell, or they can even leave the cell through exocytosis. And we can use certain components of our cytoskeleton in order to allow these secretory vesicles to travel, because the cytoskeleton itself is really the bone and muscle of the cell that is going to be made up of protein filaments like microtubules and actin filaments that will extend and help these vesicles move around. All right, here we have our lysosomes. These are sac-like structures that are found throughout the cytoplasm. They contain enzymes in here so that they can digest either pathogens or certain components that the cell no longer needs. And if a cell were to become injured, that could cause a release of these enzymes that can lead to cellular self-destruction. Something that people get mixed up with our lysosomes are the peroxisomes, and these also contain enzymes, which is why I think people mix them up. These are oxidative enzymes that help to break substances down into harmless products. So I would say the biggest difference between our lysosomes and peroxisomes is that our lysosomes are more responsible for breaking down waste products inside our cell using digestive enzymes, whereas our peroxisomes are more used on in oxidative reactions, such as breaking down our fatty acids and detoxifying harmful substances by producing hydrogen peroxide and then converting it to water using an enzyme catalyst. So again, water being harmless. All right, next we've got our star of the cell, I like to call it, which is the mitochondria. And these are used for cellular energy metabolism, essentially producing ATP, giving us energy and our cells energy. And it also has a role in osmotic regulation, pH control, calcium homeostasis, and cell signaling. We're going to touch upon some of these topics as we move through the lecture. We've already kind of talked about the cytoskeleton, but just wanted to show you these structures like the microtubule, actin filament, and the intermediate filament. They're all kind of working together to help give this cell its shape and also allow substances to kind of ride along these different filament types. Okay, hopefully this is an easy question for you. We have the function of cellular energy metabolism and generation of most of the cell's adenosine triphosphate, or ATP, occurs in the, and hopefully you chose the mitochondria. We know that the nucleus is going to play more of a role within cellular division and control genetic information. Our lysosomes are going to be those sac-like structures that are going to have the digestive enzymes within it. And then the Golgi complex is the one that is going to process and package our proteins into secretory vesicles. So that leaves us with the mitochondria that is known for producing or going through our cellular energy metabolism to generate most of our cells' ATP. Now, another big structure of the cell is the plasma membrane, and this is really important because it's truly the boundary between what's outside of the cell and what's inside of the cell. So it's controlling the composition of a space or compartment that it encloses. Let's talk a little bit about the functions of the plasma membrane. So first we have our cell-to-cell recognition, which allows a cell to recognize another cell, and we usually use the carbohydrates along the plasma membrane in order to do this. We also have cellular mobility, which allows a cell to move from one location to another, and it's really critical in certain biological processes so that we can coordinate mechanisms that involve the cytoskeleton skeleton, our cell membrane, and motor proteins. It helps maintain our cell's shape. We see that the plasma membrane is typically thicker than that of our intracellular organelles, and so it'll help to contain those organelles and also maintain the relationship with the cytoskeleton, our ER, and other organelles, as well as maintaining the fluid and electrolytes within the cell and outside of the cell. And lastly, we have the movement of our molecules. This will help our contents be able to communicate with one another. We will also have some anchors known as integrins. We'll be able to release certain enzymes when needed, and our antibodies to the extracellular environment, and also help to create a symbiotic, nutritive relationship amongst these cells. When we look at the plasma membrane's composition, we see that we have this lipid bilayer here. The molecules within here are known as phospholipids, and if we look at the very top, we could see that they have a polar or hydrophilic head, meaning that they love water, so they can face the water outside of the cell or inside of the cell. And on the internal area here, we have our two fatty acids, and these are the nonpolar hydrophobic portion of our phospholipids, also known as the tails, whereas this is our head. Now, though we have this structure in place, our plasma membrane can change its physical state depending on the temperature, our lipid composition, and presence of other molecules such as cholesterol. And so these three different states are going to be called the soft solid gel phase, the fluid liquid crystalline phase, and liquid order phase. With our solid gel phase, this is going to be when our lipid molecules are tightly packed together, they're really rigid, and our fatty acid chains are fully extended and immobilized. So this really is going to create a stiff plasma membrane that's less dynamic. With our fluid liquid crystalline state, we'll see that our lipid molecules are more disordered, so we'll have increased mobility of our plasma membrane, and our fatty acids are going to be more flexible and will allow more lateral diffusion of lipids and proteins. And lastly, we have our liquid ordered state, and this is going to kind of be a combination of the first two states. So our lipids are pretty ordered, and they are going to have some lateral mobility, and so our state is more stabilized if we have that presence of cholesterol, and that helps to organize our lipids and maintain fluidity. And so our phospholipids are known to be amphipathic, which means that they have that hydrophobic portion of the tails and the hydrophilic portion of the head. And with this composition, we are going to have it serve as a barrier to diffusion of water and hydrophilic substances. So it'll allow the lipid-soluble molecules such as oxygen and carbon dioxide to diffuse through the membrane more readily. And our lipids, along with protein assemblies, are going to act as molecular glue for the structural integrity of our membrane. Now let's talk about the proteins associated with our plasma membrane. They're going to perform most of the tasks here. We've got lots of functions that they assist with. So let's start off with their function as receptors. They are going to bind to specific molecules that initiate a cellular response. As transport channels or carriers, we don't really see those channels pictured here, but they'll help facilitate movement through substances across the cell membrane. They also serve as enzymes that'll speed up chemical reactions. Surface markers is exactly what it sounds like. They're proteins that are going to help identify cells and mediate an immune recognition. And then we have our cell adhesion molecules known as CAMs. They will help stick to each other and their surroundings, hence the term adhesion. And then our catalysts. These are our proteins that'll accelerate our biochemical reactions without being consumed themselves. And here we see the different types of proteins that we can have. In A, we see transmembrane proteins, trans meaning through, so we see them sitting through the plasma membrane. We can have a single alpha helix, so that coiled conformation, or we can have several of those helixes, multiple alpha helices, I should say here. And then we have our beta barrel as an example. We can also have it as a monolayer associated alpha helix, and this means that they're anchored to the cytosolic side. Here's our cytosol here inside the cell. And they are an amphipathic alpha helix here. In C, we could see that they are kind of implanting themselves into the plasma membrane by having a covalently attached lipid molecule. So that's why it says lipid linked. And then lastly, we have our protein attached, which means they're attached by weak non-covalent interactions to other membrane proteins within the area. So let's talk about how we regulate these proteins, also known as proteostasis. This is essentially a state of the cell that balances the processes of protein synthesis and folding and dehydration. And you can imagine that we are going to have to adapt depending on how quickly these proteins are made, how long they're surviving, and when they break down. So we have some proteolytic systems that assist us in doing this so that we don't misfold or have protein aggregation take place. We are going to regulate these processes by using the ribosomes. They're going to help make our proteins, and the chaperones will help with this process. And like I said, we've got two proteolytic systems. So our lysosomes are going to assist us with this. You can kind of see their role over here. And our ubiquitin proteasome system, also known as UPS, which we can see at work over here when things are misfolded so that we can correct the situation. And whenever we do have any malfunctions of aggregation or misfolding, that can lead to disease. So now let's touch upon our carbohydrates. They play several roles within our plasma membrane too. As far as protection, we have our glycocalyx, which is going to be a carb-rich layer that surrounds the cell and really shields it from physical or chemical harm. We have lubrication because our carbs are going to have a mucus function that serves as a lubricant that minimizes friction on the cell surface in areas like our joints and our digestive system. Then we've got recognition. I mentioned this earlier, where we're going to have markers such as our glycoproteins and glycolipids that will serve within our immune responses and signaling. And lastly, we've got adhesions, which the carbs help to facilitate these cells coming together, binding them to one another to support tissue formation and our integrity of these cells. Now let's move into cellular receptors. We call these ligands just as a general term, and the word ligand actually means to bind. So they will bind with a cellular receptor. I liked this simplified image here where you see the ligand coming close and binds over to the receptor, and that will activate or inhibit the receptor's associated signaling or biochemical pathway. We discussed how we have some plasma membrane receptors, and they determine the response to the binding. So for instance, we can have that ligand be a hormone and attach to our receptor, or we can have a protein that is going to be a channel-like receptor. This is a non-channel-like linked receptor, also known as a ligand-gated channel. So sometimes when the ligand attaches, the channel will open. It'll basically kind of unlock, which is what we're seeing over on this side. And then over here, we are seeing an example of a ligand-gated ion channel where we are going to have a neurotransmitter attached to a special spot on this receptor, and that then will cause this channel to open up, allowing a ion, not that same neurotransmitter, but the ion, to move through. Next up are our cell-to-cell adhesions. These are formed on our plasma membranes and are going to be held together by an extracellular membrane, cell adhesion molecules, or specialized cell junctions. So we know that our cells are soft and flexible, and we discussed how their cell integrity is based on their membranes for survival. So sometimes we have to have these strong connections between cells in order to form tissues and organs and allow them to have important functions like muscle movement. So let's talk about some of these adhesions. First up is going to be our extracellular matrix, which is secreted by the cell itself. We know that we're going to have our fibrous proteins that sit within this gel-like substance, and this is all produced by these cells known as fibroblasts. And it helps to produce a meshwork that is going to have two main classes of macromolecules that would include our proteoglycans, which you could see pictured over here, and they're bound to our polysaccharide chain, and our fibrous proteins, which would include collagen, elastin, and fibronectin, and laminin, which have structural and adhesive properties. And through this network, we are going to allow for diffusion of nutrients such as water, and things like lipids, also some metabolites and hormones between our blood and our tissue cells. And this extracellular matrix will also help to regulate cell growth, movement, and differentiation. What about the basement membrane? We saw that in this image over here, and a closer look at that basement membrane, which has a specific type of collagen in there, a type 4 collagen. There are many different types, but the most well-known are 1, 2, 3, and 4. And so our basement membrane is more of a specialized type of extracellular matrix, and it creates these sheets in which we can attach cells to. They could be thin, tough, or flexible. And areas where we're going to find them is beneath an epithelial layer of cells. So right over here we see these more columnar, simple columnar epithelial cells sitting on a basement membrane, which is also known as the basal lamina. We can find them between two sheets of cells like we do within the kidney's glomerulus. You could see the endothelial cells on either side of the basement membrane. And then we also have it surrounding individual muscle cells. These happen to be our smooth muscle cells over here, but they can also surround fat cells and our Schwann cells, the ones that make up the myelin sheaths for the axons of our neurons. Okay, and here is probably what you hopefully thought of when I said cell junctions. These are our specialized cell junctions, and we can classify them as symmetrical or asymmetrical. So first up are the tight junctions, and we see that illustrated over here. These are the tightest junctions that we could have. They create a seal between two adjacent cells, which would prevent the passage of molecules and maintain distinct environments on either side of the tissues. We would find this in areas like the epithelial cells that line our intestines and also our blood-brain barrier, especially within the brain, so that we are preventing leakage. Next up are our desmosomes, or our belt desmosome, which you could see pictured over here, and they are going to work to anchor the adjacent cells together, and they connect the cytoskeletal filaments, which is kind of why we see these lines here representing the filaments, and they help to provide mechanical strength to our tissues. And so we want them in areas that are subjected to mechanical stress, like our skin, the epidermis, and our cardiac muscle. Then we have our gap junctions, which is going to help us with communication. These are going to form between two neighboring cells that allow an exchange of ions and small molecules. Again, more for communication. So areas we'd want to see them in is like our cardiac and smooth muscle, where we're going to want rapid transport of our ions and also those electrical signals so we can have synchronized contraction. These junctions are made of something called connexin proteins, and that will influence their gating properties. Next, we have the asymmetrical type, which is a hemidesmosome. You could see some of these placed along the bottom over here, and these are helping to anchor these cells to an underlying tissue and maintaining structural stability. So again, we can find this in areas like the skin, where we want to take a basal layer and attach it to a basement membrane. So I mentioned the connexins are going to help us with gating properties. This is referring to the process of controlling the opening and closing of the gap junction, these channels that get created, or pores between the two cells. And so typically we find this always associated with the gap junctions, like I said, and they're controlled by the pH volume, voltage, calcium ions, and the amount of connexin composition, so the amount of proteins we have creating those pores. And this enables our uninjured cells to protect themselves from the injured neighbors. Next, we have cellular communication. We know that our plasma membrane is going to assist with this through these bound receptors that we have on the plasma membrane. So this is what we would call more of a direct physical contact between cells. We are also going to have, pictured over here, remote signaling by secreted molecules, and this is really representative of our paracrine signaling. We're going to talk more about these different types of signaling on the next slide. Actually, let me just kind of move through our last type of communication that we have, and that is through our gap junctions that I mentioned earlier, where we are able to pass those ions back and forth. If a cell cannot perform this gap junctional intercellular communication, then normal growth control and cell differentiation is going to be compromised, and this could lead to cancer tumor development. So let's talk about these different types of chemical signaling. So we have our contact dependent signaling. This is where we have that membrane to membrane contact. Paracrine is what we saw in the previous slide. Basically, we have a cell here that is secreting a substance that travels to other cells. Typically, that's a different cell type than the original one, and gives off whatever chemical or hormone it is secreting. Then we have autocrine. Remember that auto means self, so that our autocrine signaling means that we are secreting a substance and it is acting back on itself. And actually, this autocrine signaling is used a lot by cancer cells so that they can stimulate their own survival and proliferation. Then we have hormonal signaling, and this involves when we have a endocrine cell that is going to secrete chemicals called hormones that are put into our bloodstream so that it could travel to a farther target cell and trigger a certain response. And then we have our neurohormone secretion. So here, our neuron is going to secrete a chemical, or hormone rather, that is being put into our bloodstream and then carried over to its target cell. Whereas when we think of our normal neuron signaling, we're going to have that nerve cell secrete a neurotransmitter that is going to be within short range and work on its target cell. And typically this is going to be a cell that the neuron is innervating to cause a specific reaction. Our next topic is signal transduction. So first we talked about cell-to-cell communication. What happens when we get that chemical or ligand, whatever it is, to our target cell? So what happens is that we are going to have it attached to a receptor, and that is going to provide some type of signal to activate a protein kinase, for example, and allow the cell to grow and reproduce, die, or differentiate. So there are, on a longer plasma membrane, 3 classes of, let's say, receptors that are going to produce some type of this signal transduction. So we have our channel-linked or ligand-gated ion channels. We also have our catalytic receptors or enzyme-linked receptors, and these could be activated by ligands as well and function directly as enzymes. And then we have our G protein linked, which is going to create an interaction with our GTP, our guanosine triphosphate, which binds to these different subunits over here to trigger the response. So by a ligand attaching to these types, we are going to activate a signal transduction pathway, which are communication pathways that could cause a cascade effect. Now, if a cell is not really getting these signals, a lot of these cells will undergo cell suicide, known as apoptosis or programmed cell death. And so what we're seeing over here is a signaling process where the ligand's attaching to the receptor, and then we are going to have that cascade take place and amplify and distribute it in its transit. This amplification is often achieved by stimulating enzymes, and depending on what this ligand is supposed to do, we could have these different cell behaviors as a result. So we could have them survive, differentiate, grow and divide, or die, like we said, through apoptosis. And so through that process of signal transduction, our first step is to have that ligand bind. And so we call this the first messenger. And then that receptor, when it's attached to it, we are going to have reception, and that could cause an opening and closing of specific channels and regulate the flow of ions into or out of the cell. Then comes transduction, where the signal is transferred to an intracellular messenger, which we can call our second messengers. If you recall, we talked about this in the endocrine system and said our second messenger was our cyclic AMP, adenosine monophosphate, and certain times it can be calcium. And then we have induction that takes place, where that second messenger is initiating the cascade response within the cell. Shifting gears again, let's talk about cellular metabolism. This is going to include all the chemical reactions that take place within a cell. We know that we have 2 different types. We have anabolism, which is energy using, and ana means upward. So we're kind of taking the smaller molecules and building up to a larger molecule so we can build cellular structures. The other process is catabolism. Remember, think C for cut, A for add. And so with catabolism, this is an energy releasing process. And kata means downward. So we are going to cut these bonds apart and release them down into their smaller subunits. And in that process, we're releasing energy. Our metabolic pathways will help us produce ATP through multiple steps, and these reactions are directed through enzymes. So we'll see some of that taking place when we talk about how we make ATP. But before we get into that, let's talk about what ATP is. Again, it stands for adenosine triphosphate. We could see the chemical compound over here, and it's really working as a universal fuel inside our living cells. It helps to drive biological reactions for cellular function, and is going to transfer and store energy through the metabolic processes. So we know Just for example, in glucose metabolism, if we have 1 mole of glucose and oxygen is present, we are going to drive that reaction forward and get carbon dioxide, water, and 686 calories of chemical energy. Just to give you an idea of what 1 molecule of ATP can give us. Now, we also know that sometimes we can break off these phosphate bonds here and we can release some energy. So if we were to break off just 1 phosphate here, we would then get our ADP, adenosine diphosphate, and we would release 7 calories per mole. Now let's dive into cellular energy. We're going to talk about this as 3 different phases. And in phase 1, we are going to begin with food. So we are going to use the catabolism, the breaking down of our proteins into amino acids, our polysaccharides or carbs to break down into simple sugars, and our fats to break down into fatty acids in order to drive this reaction forward. So these nutrients are hydrolyzed within our intestinal tract. We absorb them and then circulate them and use them for ATP production. All right, so in that first phase, we are going to have digestion take place. We got to break these into smaller components that we just named, right? In phase 2, we are going to want to utilize the process of glycolysis. This means we are going to take up our simple sugars like glucose and in certain instances some amino acids to achieve pyruvate, 2 molecules of pyruvate to be specific, and we can get 2 molecules of ATP per glucose molecule that we used at the beginning of glycolysis. And our next step is going to be to take that pyruvate and convert it into acetyl-CoA. Now this process limits the ATP that is produced, but we have a couple mechanisms that can maybe drive up our amount of ATP. That brings us to our third phase, which is going to be where we have our citric acid cycle and oxidative phosphorylation take place. This citric acid cycle is also known as the Krebs cycle or the tricarboxylic acid cycle or TCA. And when oxygen is present, it can drive us into oxidative phosphorylation and we can get a lot of ATP produced. We also have waste products that are excreted. Now the way we're going to do this is we are going to reduce our NADH, our reduced nicotinamide adenine dinucleotide, and also our reduced form of FADH2, also known as flavin adenine dinucleotide, both of which are helping transfer their electrons into an electron transport chain so that we can eventually get to our final electron acceptor and pump out a bunch of ATP. And so let's take a look at that oxidative phosphorylation process a bit more. It's going to take place within our mitochondria. So just to draw your attention to this figure over here, first in the cytoplasm, we are going to have the process of glycolysis take place, right? So here's our anaerobic process versus the aerobic process, meaning here we don't have oxygen, here we do have oxygen. So in both, we're going to go through similar processes where we produce our 2 ATP. When we have our, let's start with our aerobic process, we are going to convert pyruvate into pyruvic acid, and I'll show you that conversion in the next slide. And then since we have oxygen present, we go through the Krebs cycle, can produce 2 more ATP, and then we can go through with our mitochondrial enzymes here in the mitochondria and go through oxidative phosphorylation in order to create a massive amount of ATP. I just want to show you this image down here. This is showing you the intermembrane space within our mitochondria, and in oxidative phosphorylation or electron transport chain, we're going to see these 4 different cytochromes is what they're called. And here they are iron-containing proteins that are going to accept the electrons, which will then combine with oxygen and release hydrogen ions and produce ATP. Now let's talk about the anaerobic process. If oxygen is not present, then we are going to shift pyruvate to pyruvic acid. So you can see here we have that hydrogen that's added on. And from there, we will shift into our lactate or our lactic acid, depending on if we're starting with pyruvate or our pyruvic acid. And so you could see the change taking place over here, where with our lactate, we're going to add 2 of our hydrogen ions. And so when we do this, we are not moving into the Krebs cycle, and so our ATP is limited. But once oxygen is present, we can convert everything back to pyruvate and move into our Krebs cycle and have this whole process take place to make more ATP. Okay, another big topic we discussed in the past is membrane transport. Essentially, this is our cellular intake and output. Our cells are always taking in nutrients, fluids, and chemical messengers from their extracellular environment and expelling their metabolites or products of metabolism, and also any of the end products of lysosomal digestion. So 2 ways we can go through transport that we see pictured here, which would be more of a passive transport, meaning the movement of molecules are moving down their concentration gradient without ATP being present. We can have our transporters or transporter-mediated transport. This has specific binding of molecules to a carrier protein. And you could see an example of this over here as well. And so as that molecule is going to attach, we have that protein undergo a conformational change. And that's better illustrated up here, where we're having this solute attach. We get the conformational change. So maybe that solute can move out of the cell, for example. Examples of proteins that do this would be our glucose transporters, or GALT, G-L-U-T, in order to transport glucose, and also our amino acid transporters. The other type we see pictured here are the channel-mediated transporters, like we see pictured over here and also over here. And these, we will see a movement of the ions or water through a channel protein. And this really allows for rapid transport of things like ions like sodium, potassium, calcium, and also you may recall the aquaporins, which allow water to move through. And then this is just a little table I put together here to show you some differences between the 2 types of transport. Again, these are both passive. Other passive transports would be our osmosis, hydrostatic pressure, and diffusion. And when we compare that to our active transport, we know that we are moving against our concentration gradient. So it's kind of more uphill. It's going to require energy or ATP. And examples of active transport would be pumps, endocytosis, and exocytosis. So just to recall some of our knowledge on this topic, we can see here some active transport where ATP is attaching. We've got the greater concentration here, lower concentration here. So this means that we are trying to move from a lower concentration area to a higher concentration area, which is opposite in passive transport, where we are seeing movement from a higher concentration gradient to a lower concentration gradient. We see some pumps pictured over here, and this is going to carry out active transport in 3 different ways. We have our coupled pumps that are linked to that uphill transport moving against that concentration gradient. We have our ATP-driven pumps that essentially are using hydrolysis of ATP, breaking it down to ADP and an organic phosphate to move against its concentration gradient. And then we have the light-driven pump, where we are going to find mostly within bacteria and use the energy of sunlight to drive this type of transport. And then other examples, like we said, are endocytosis, where we're moving the contents into a cell, and exocytosis is moving out of a cell. And here's some mediated transport processes where we're moving solutes singly or two at a time across the membrane. So first example listed is our symport, sim meaning two or same, where we're moving in the same direction, even if they're two different types of molecules. Then we have antiport, anti meaning against. So these contents are moving against one another, meaning in different directions. And then we have uniport, where we have a single molecule that's moving in one direction. We also have our electrolytes, which make up about 95% of the solutes within our body. An electrolyte means that it is electrically charged, so it's going to dissociate into ions in a solution, and they exhibit polarity, which is what we see over here. We have our cations pictured in green, meaning they're positive and they would migrate to a cathode or a negative pole, because positive is attracted to negative. And then we have our anions that are negatively charged, so they would migrate over to a anode, which is the positive pole. And we have to have a good balance of these within the body, because we are going to utilize them for nerve and muscle electrical impulses. Just to give you some examples, our sodium, like we saw over here, is positively charged. It doesn't want its electrons, so it could be stable. If you remember that valence shell, we want the octet rule to be fulfilled, so it is going to want to give its electron away, whereas chloride, that needs one more electron to fulfill the octet rule, will gladly take it. And so chloride becomes our anion and our sodium becomes our cation. And you may have already noticed through some of your courses that we measure electrolytes in milli-equivalents per liter. So here's our units for that, and also our milligrams per deciliter in this unit written over here. And then just some terminology here regarding its charges. So if we just have one charge, like our sodium ion, then it is called a monovalent. Whereas if we have two charges, like calcium, for instance, we would call that divalent. Okay, a bit more on passive transport. Again, this should be review for you. So simple diffusion is where we have that movement of solutes from a greater area of concentration across the plasma membrane to an area of lower concentration. Energy is not needed here, and it could be influenced by the size of our molecules and lipid solubility. Filtration is a little bit different in that we are going to see movement of water and solutes through a membrane. So this would serve as our membrane, and that is because of a greater force that we are having on one side. So for instance, this is before we move across the filter. So we see that we have lots of solutes here. The red molecules here are too big to move through the filter, and so the small ones will move through, and then after we're going to have the molecules that were retained. So maybe we'll have some of those little small yellow molecules, but we'll definitely have all of those red ones. And this really reminds me of how the blood kind of flows through the glomerulus in the kidney, where we're going to have those smaller contents and water move through here, but those larger plasma proteins within the blood and our red blood cells, that's all going to stay over here and move into what would be the efferent arteriole, right? We recall that, I hope. And then down here we see our figure illustrating our hydrostatic pressure and oncotic pressure in the plasma. And so at the very top, we see the weight of our water that's creating a hydrostatic pressure within our plasma as the blood moves through. Whereas down here, we see demonstrated the oncotic pressure, and this is the pressure that is exerted through our proteins within the plasma. So when we have large proteins here in our plasma, it tends to want to pull water into our blood. So if we don't have those proteins within our blood, maybe it's due to starvation, we're not able to make those proteins and have them within our blood, then we are unable to keep that oncotic pressure, which means we're not going to be able to absorb water into our bloodstream, and that could cause edema within our body. We keep those fluids within our tissues because we're not pulling it into our blood. All right, and then we have osmosis, again, a very simple concept that we've talked about before. And this is when we have the movement of water down the concentration gradient. So we have our beginning over here where we have high concentration of solutes on this side. We know that water is attracted to solutes, so it is going to want to move through that membrane and over to the right side. And this helps to control the distribution of the water within our body compartments and equalize our fluid within our intracellular fluid and our extracellular fluid. And it really is reflective of our hydration status. And so that osmosis or movement of water is related to our hydrostatic pressure and solute concentration. And I think one of my favorite images to illustrate that is this over here, where we're talking about our movement of fluid as we look along a capillary. And so here we could see on the arterial side of the capillary, we are pushing the fluid out here. We see that our capillary blood pressure is high here. And then as we move over to the venule side, we're going to see that the fluid is actually starting to be pulled back in. And that is because we start getting a decrease in that capillary blood pressure. Another example of that in the capillary is shown right over here, where we have that hydrostatic pressure helping to push the fluid out, whereas as we move closer to the venous end, we're going to start to have that osmotic pressure play a larger role to bring it back in. And then over here, we have our interstitial fluid in this area. Here's our intravascular fluid. And so we can see the hydrostatic pressure once again pushing it out. And then as we move down, we start to change that and drive the fluid in. Another thing to note is going to be our tonicity of our cells. This is important, especially for our red blood cells. Here we see an isotonic solution where fluid is going to be able to shift back and forth. We keep the natural size of our cell. Whereas over here in B, we see the red blood cell placed in a hypotonic solution where we have solutes higher inside the cell, and so water is going to be driven inside of the cell, and the cell will lyse. It'll swell first, and then it could possibly lyse if it pulls in a little too much water. And then we have our hypertonic solution where we have lots of solutes in the solution, not as many inside. So that drives the water out of the cell, which causes the cell to shrivel or have crenation. So we've used the terms osmolality and osmolarity before. Let's talk about the differences here. It's really just a way of measuring the concentration of our molecules in different ways. So with osmolality, this is going to be a measure of the solute concentration by its weight. So we're using kilograms over here to do this. Whereas osmolarity, we are measuring the solute concentration by volume. And that volume is that we're using is liters. So what you can expect in clinical practice is that the preferred osmolality in clinical assessments is going to be between 280 to 294 milliosmoles per kilogram. And so I just like to think that we want that number close to 300 if you want something simple to memorize. And now we have to shift into active transport. We use a system for sodium and potassium. If you remember that sodium-potassium pump pictured over here, we find this pump in all mammalian cells. And it really is an antiport system, right? We are going to move those 3 sodium ions out of the cell and move 2 potassium ions into the cell. And we use ATP to kind of fuel this process to take place. And the reason we have this pump in our body is to generate an electrical potential, making the inside of the cell more negative. So we are going to use 60 to 70% of our cellular ATP, especially within our nerve cells and muscle cells, to do this. And here's another question for you. Active mediated transport is going to involve which one of these? Okay, we know that diffusion is really passive transport. Moving down a concentration gradient is going to be a passive transport. And then what do we think? The mechanical force of water pushing against cellular membranes. That kind of sounds like moving down the concentration gradient as well, right? So our answer is expenditure of metabolic energy because we use ATP for active mediated transport. Some other transport processes that we mentioned earlier are endocytosis and exocytosis. In endocytosis, we are going to have that vesicle form and move into the cell. If it is pinocytosis, we are bringing in fluids. If we're bringing in large solid particles, then that process is called phagocytosis. You may remember these as cell eating and pinocytosis as cell drinking. And then these can also, especially with our pinocytosis, involve clathrin-coated vesicles to bring that fluid in. With exocytosis, we are going to have the opposite kind of take place. We're going to be secreting macromolecules that are synthesized in the cell and leave or take some of the, release some of those contents outside of the cell through the plasma membrane and move into the extracellular matrix. Now, both of these processes are going to require ATP and will often involve the plasma membrane receptors. You may also remember our resting membrane potential and action potential. The resting membrane potential is when we have lots of potassium inside the cell and lots of sodium outside of the cell. And then as we go through the process of depolarization, we are first going to have a stimulus that causes the sodium channels to open up, and we start to get sodium moving inside of the cell. This drives that resting membrane potential up more positively, up until about 0 millivolts, and so it's becoming positive, right? In order to have the action potential take place, we have to pass a threshold, and that could be something like -55 millivolts. Now, sometimes we don't reach that threshold, and we can have some hyperpolarization taking place where the membrane potential is more negative than normal. So let's say our resting membrane potential is -70. If we were to go down to -75 or -80, that would be hyperpolarized. Whereas and so that would be, let's say, decreased excitability. Whereas if we have hypopolarization, then that means we are a little bit more positive than normal. So again, if our resting membrane potential is -70 millivolts, then maybe this is -65 millivolts. So we have increased excitability. You may also remember this being referred to as an EPSP within our neurons, and that can help drive it closer to the threshold, which if we reach the threshold, then we get depolarization that takes place, leading to our action potential. And then once we reach that action potential, we have to have repolarization take place, where we are going to close those sodium ion channels and open up the potassium channels so that we could start bringing in the potassium into the cell and restoring the negative polarity. We'll also have that sodium potassium pump helping us put all the ions back into their proper space. Now, during that period, we are going to have something called a refractory period. And that essentially is when if it's absolute, it's when our membrane is unresponsive to any other stimuli, meaning we cannot achieve an action potential. When we have a relative refractory period, that means that we need a very strong stimulus, more than normal, in order to get an action potential to take place. And that moves us into our cell cycle. Now, as humans, we produce millions of cells per second in order to sustain life. We have to have cell growth and division in a repetitive sequence known as the cell cycle. And this continuity of life is going to depend on this constant cycle of duplication and division. So we have a couple processes. Again, you may remember these. We have meiosis, which is specifically a reproduction of our gametes, of our sperm cells and egg cells. We talked about this at the very end of ANP2. With mitosis, this is the division of our somatic or body cells into two identical daughter cells. And so we have these two sequential phases that take place in mitosis. Mitosis is the actual nuclear division, and then cytokinesis is where we're going to divide that plasma up as well, and we get these two distinctive cells. Now, most of the time we have to go, not most of the time, all the time, we have to prepare for this division to take place. And to do this, we have to go through a phase known as interphase, where we have the growth phase of the cell where it can double in mass, duplicate its DNA content and its organelles, and also a lot of our cells will spend majority of their time in this interphase. So let's talk about the 4 phases of our cell cycle. We've got our G1 over here, which is the period between mitosis and the start of DNA synthesis. When we go into our S phase, this is when we are synthesizing our DNA. And then our G2 phase is going to be when RNA and protein synthesis take place. And lastly, the M phase is where mitosis is happening, where we're actually getting cellular division. Now, this cell cycle is regulated by something known as cyclins. And the cyclins will have levels that will rise and fall depending on what is going on in this cycle. And our cyclin-dependent protein kinases, or CDKs, are assisting as well. And we've got inhibitors that work through the cycle as well. So let's review our M phase, our mitosis phases. So we know that first we have to have interphase take place. It's our longest phase, includes G1, S, G2 phases. And at that point, our chromatin is long and have slender rods. They're jumbled in the nucleus. And then in late interphase, we're going to have that chromatin coil up and become shorter and thicker. So they condense. So prophase is going to be our first phase. You could see the early phase of prophase here, and this is when our chromosomes first appear as sister chromatids. So you can see them lined up next to each other, and they're joined in the center at something called a centromere. You can notice that our nuclear membrane is disappearing, and also our spindle fibers or asters are forming so that they can attach to the chromosomes. You can see them lengthened kind of over here in this late prophase. Then we are going to start to align them down this equatorial plate or the middle of the cell in metaphase, and our chromosomes line up and are highly condensed and are easily observed under the microscope. Then in early anaphase, we will start to see that the centromeres are going to split and the sister chromosomes are being pulled apart to opposite poles. And the cell is going to briefly have 92 chromosomes, which separate into 2 groups of 46. And here's the later phase of that where we see them traveling more to the opposite poles. And then we are going to have telophase, where we start to get the appearance of a new nuclear membrane around each of the 46 chromosomes. Those chromosomes begin to uncoil. We could see that better in this late phase over here. And actually, I should have called this a late phase. This is really our cytokinesis taking place, because at this point we see that cytoplasm has split, creating 2 identical diploid daughter cells. And then over here, this is just an image or a micrograph of a onion root tip that's showing the different stages of mitosis. You can kind of see the chromosomes lined up here. That's a good observation of metaphase. Here it looks like they're the chromosomes are pulled to either side of the pole. So this would be like anaphase, for example. And then how do we control cell division and growth? This is a complicated process, but the key processes are going to include cell growth, division, and survival. These processes are regulated by intracellular programs and extracellular signal molecules. Some of those are listed over here. And I didn't want to read through all of this, but let's focus on our mitogens. These are going to be chemical agents that are stimulating mitosis. They act as extracellular signals from the neighboring cells, and their function basically is to stimulate growth, differentiation, migration, and survival of these cells. Our growth factors, which could be referred to as our cytokines, also help stimulate cell growth. They're going to promote protein and macromolecule synthesis and inhibit protein breakdown. And lastly, we have our survival factors. That is going to inhibit programmed cell death, so apoptosis, in order to allow that cell to survive. Lastly, we're going to talk about tissues. So you may remember that we have 4 principal types of tissues within our body. We've got our nervous tissue, which is highly specialized cells that would be our neurons, but we of course have our glial cells in here. We've got our epithelial tissues that cover most of our internal and external body surfaces, and then connective tissue that will bind our tissues and organs together. And lastly, muscle tissue that's going to be composed of our monocytes or muscle cells that enable movement to take place. There is a table in your book, table 1.6, that gives you the specific types of each one. So I'll just choose the simplest one, muscle tissue. It goes through and talks about your cardiac, skeletal, and smooth muscle tissue. So if you want a refresher on all those tissues, I highly suggest you go and visit that table. Now, what about tissue formation? We are going to have intercellular recognition and communication, adhesion, and memory. Basically, we have specialized cells that sense signals like growth factors to produce new cells when they're needed. As far as the adhesion, this is when our cells are selectively adhering to other cell types that are the same, or maybe to extracellular components. And the strength is provided by that extracellular matrix, our cytoskeleton, and the cell-to-cell adhesions that we talked about. We, as far as the memory, our cell is going to maintain the unique characteristics through gene expression during embryonic development, and this memory is passed to progeny, ensuring that we have tissue identity. Now, first, let me talk about our stem cells over here. These will go through self-renewal and divide indefinitely and maintain their population. If we have multipotency, it generates all differentiated cell types within their tissue. But sometimes we can have a differentiated cell that is terminally differentiated and non-mitotic, so they are no longer able to almost like reproduce themselves. And down here we can see something called WNT signaling that is fueling and progressing tissue renewal and stem cell activity in diverse organs such as the brain, skin, intestines, and the mammary glands. This is a complex cell communication pathway that plays a critical role in embryonic development, tissue homeostasis, and other cellular processes, including cell proliferation, differentiation, and stem cell renewal. So we can use these in things like cancer, neurodegenerative disorders, and developmental defects.